much for the introduction. I'm really excited to see you all and wish I could be there in person. I know that the wildflowers must just be popping off right now, and Southern Illinois holds a really special place in my heart. So, as you said, I'm a PhD candidate at the University of Montana. But today I'm going to be discussing research that I conducted in Dr. Jen Weber's lab in the genus Triodanis, which is commonly known as Venus's looking glass. So my talk is 100 years of phenological sensitivity in Venus's looking glass. So Venus's looking glass, commonly called Triodanis, this is a genus of 7 species that are native to the Americas. And here are just 3 of them. We have Perfoliata, Lamprosperma, and Biflora. There's 7 species in total, and all 7 Triodanis species are found in Texas. 2 species, T. coloradoensis and T. taxana, are actually found only in Texas. But then on the other end of the spectrum in the genus, T. perfoliata, or clasping Venus's looking glass, is found throughout the Americas. So this is a map from Plants of the World Online that show, all of the places in the 2 continents that it's found. And that also doesn't count areas of Asia where it actually is introduced as well. Clasping Venus's looking glass, it's just an example of something that has really thrived in weedy, human-disturbed areas. Here you can see it growing on the roadside. Here you can see it growing out of a crack in some asphalt in someone's garden. And its kind of weediness and existence as like a sidewalk crack plant, is probably part of why it's found. It's so ubiquitous. So I'm going to call it like Perfoliata or T. perfoliata for the rest of this time. One thing that makes Triodanis really interesting from an evolutionary and ecological perspective is its mating system. And so about like 10, 15 years ago, not too much was known about this wildflower genus, but Dr. Weber has really been laying the groundwork and developing it as a great model system for plant evolution and ecology. So let's talk about the mating system. It has what's called a mixed cleistogamous mating system. And what this means is that it has these outcrossing or chasmogamous flowers. So chasmogamous, think of like chasm, like open. And so these are, they just look like the regular flowers. They're the ones that have the potential to outcross. But it also has these cleistogamous or CL flowers. These are closed and they don't have petals. They don't really look like a flower, but inside they have the male and female reproductive parts. They can produce seeds, but those seeds will be completely self-fertilized. And a single individual plant will have some mixture of the CH open flowers and the CL flowers. And when collect these plants, you can count each type, and this can be used as a proxy for the selfing ability of a particular individual. And this ratio of CH to CL, it can vary a lot between individuals, between populations, and even across the landscape. And so for a little bit more background on this, I'm kind of building on some of the work of Dr. Beth Ansaldi, who did her dissertation research. She was advised by Dr. Jen Weber. And among many other things that Beth did, she conducted this extensive field survey of Triodanis perfoliata from all these populations across the US and quantified the ratio of CH to CL flowers in each of these populations. And what she found was extensive variation in that ratio of how many outcrossing flowers were produced relative to how many selfing flowers. And when she coupled that with spatial modeling that used climate data, what she found was a relationship between this ratio and climate variables such as the water availability and the soil type. So in short, areas that seemed a little bit more resource rich tended to produce proportionally more of those outcrossing flowers. And this fits how we tend to think about plant mating systems, where they have this mixed mating system with the option to either have outcrossing or selfing flowers. Because if you think of plants as kind of having a carbon or energy budget, they can only do so many things, and flowers can be pretty resource expensive. They need to be colorful, they can take a lot of water, a lot of pigments. And so, if resources are poor, it could be kind of smart on the part of the plant to produce relatively more of those selfing flowers. And it provides this kind of reproductive assurance when things get tough. And so, what this research is kind of showing is this sort of remarkable flexibility in mating system because there's just so much variation in how much selfing versus outcrossing that it's doing. But at the time, like back in like 2016, there still was not very much known on whether the flowering time, the timing of that open flowering, also had some flexibility in Triodanis. And why exactly would we care about flowering time? I mean, I think we all kind of know this because you guys are all here at the Native Plant Society, but we also just heard about it in Dr. Dongermon's talk. Proper plant flowering time is just totally essential for plant fitness. If a species is outcrossing, it's really important for plants to flower when there are actual mates available. So it needs to time it well with its conspecifics in the area. If it depends on insect pollination, then it's necessary to time the flowering well to where the pollinators that are around. And then finally, as I said before, flowers can be resource expensive. So it's really important to flower when resources are available. If you flower when there's just not enough water to maintain those flowers, you might be kind of out of luck when it comes to survival and reproduction. So yeah, I think we all know that timing of flowering is important. And it's also kind of a very obvious trait, especially this time of year. We're looking around and seeing things pop up. And it may seem like this pretty simple thing, but the more you look into it, the more complex it is. And again, this is similar themes to what Dr. Dongermon was just talking about. But so imagine, this is some taxon might have some particular day of year that it usually flowers. And in order to figure out when to flower on an individual level, plants are integrating a huge variety of both external and internal cues. In terms of the external cues, for spring flowering plants, many of them need to experience winter first to know that it's spring. And so they have this winter chilling, or sometimes called vernalization cue. And then there's also the spring temperatures and the amount of water available. All of these things externally can help tell the plant whether it's a good time to flower and whether they'll have a good chance of surviving and reproducing. Now, as we all know, these kinds of things like winter chilling and spring temperatures and water, these are all changing as the climate is changing. Generally, they're getting warmer and they're also getting more random. Like there's just a bit more unpredictability in what's going on. However, another really important cue for plants knowing when to flower is day length. If you flower in the spring, often plants need to hit this minimum critical photoperiod before they flower. If you flower in the fall, often plants are cued in by shortening days. And even as the climate is changing, the Earth is staying in the same position on its axis, so day length throughout the year is going to remain constant. But again, these other things, they are not constant, they are changing. So as winter is getting warmer, this day length is staying the same. Now, these are just the external cues, but there are also a variety of intrinsic cues that help shape when a plant flowers, like an individual plant's genetics, its hormones, and the age of the plant, like of the individuals. Plants need to be a certain size before they can actually flower. And so these internal factors are going to shape how a plant responds to the external factors. And because these internal factors vary so much between species, I think it can really help to take a deep dive on a particular species or a genus to try to figure out what is cueing it to flower and shaping its phenology. In particular, as in general, winters are getting warmer and winters are getting warmer and so are the springs. It's really common that plants are shifting their phenology earlier in the year. And what that means is that they're needing to deal with shorter day lengths as well. which is another kind of interesting thing. And of course, not all taxa are shifting earlier, but it is one of the kind of most ubiquitous signs of climate change where naturalists can look around and say, hey, cherry blossom season is earlier than it was when I was a kid. All right, so as I said, flowering time, it sort of seems like a simple thing, but there's quite a lot that goes into it. And we were, studying Triodanis. This is research that I conducted in Dr. Weber's lab during my master's, and we had a couple of questions about Triodanis. We knew it had this really cool flexible mating system, but we didn't know if its flowering time was particularly flexible. So we began with these few questions, like how has flowering time in T. perfoliata changed over the last 100 years? What kind of climatic cues shape that flowering time in T. perfoliata? And then, we know the climate's changing, but does this mean that Triodanis perfoliata is experiencing stress from the climate, or is it able to temporally escape by shifting its flowering time? And we were able to address these questions using herbarium records because Triodanis perfoliata has been extensively collected throughout the US over the last century. So here's an example of an herbarium record where we have the location and the date, and we can also tell that this is in a vegetative stage, so it's green, it is not flowered yet. And so by looking through digitized herbarium records, we could score its phenology as well as anchor it in its time and place. And so, yeah, as I said, Triodanis perfoliata has been sampled extensively. Here is a picture of me sampling Triodanis perfoliata back in 2017, but I was just joining this long list of many, many naturalists and collectors that have gotten it. And so this is our map of the samples that we were able to get by leveraging digitized herbarium records. And as you can see, it's a lot more abundant in the southeast. And yeah, Triodanis is just a lot more common there. It is technically present throughout a lot of the West, but just personally, I live in Montana now. I used to see Triodanis all the time when I lived in Missouri, and now I don't think I've seen it in 4 years. So anyway, it's not that our data are bad in the West, it's that there just aren't as many Triodanis out there. Yeah, so as I said, we were able to leverage a bunch of digitized herbarium records, which were made available for free through many different herbarium consortium networks throughout the US. And before I dig into what we found when we analyzed these herbarium records, I want to make a point that I think is important when discussing this. So I think herbaria are amazing. They're an absolutely amazing resource, and I really recommend that people, use them and fight for their funding so that they can continue to exist. But I do want to acknowledge that herbaria are a product of colonialism. Western science, of which I am a practitioner, and I think we all are if we're here, really has this tendency, both literally and figuratively, to take things and put them into boxes. So kind of go out into nature, bring it back, pin it to a card. But the thing is, sometimes these naturalists that have historically gone out, they have been sort of inextricably linked with the same forces that were colonizing the continent at the same time. And to give a really concrete example of this, so I used to live, in the region around Southern Illinois, and I know there's a lot of Lewis and Clark monuments and things there because it's close to where they set off for the West. And I have this quote from Thomas Jefferson here telling Lewis and Clark to make sure to note the soil and the face of the country, its growth and vegetable productions, especially those not of the United States. And Lewis and Clark were avid naturalists, and they, this is a picture of some of their herbarium specimens. And they were motivated by curiosity, but they were also motivated by enriching and increasing the power of the United States. And so as they were surveying these areas, this was also sort of assisting in the theft of these lands. And so I also want to put in my land acknowledgement. I have this list of tribes that are associated with Southern Illinois, both in the past and in the present. And the reason why I am tucking this in right now is because I really, I like doing land acknowledgements before my talks, but I've recently tried to sort of integrate them a little bit more into the talk so that it doesn't seem like an afterthought. Because one of the reasons why we're able to study these amazing plants is due to the stewardship of the native people of this land. All right, I showed you this map before. These are the occurrence records we were able to find from digitized herbarium specimens in Triodanis perfoliata. And in the top left, we have a map which shows the warming over the last century in this same era. Oh, and sorry, I should have said these records are from the 1890s to the present. And then these two maps here, they represent our spatial sampling depth and our years of phenology data. So in the bright yellow areas, we have as many as 140 years of sampling depth. So we have this really, really neat picture of Triodanis over the last century using this database. So we were interested in whether flowering time has shifted over time, and I'm just gonna jump right into what we found. Oh, but before that, I want to give one more pitch for why Triodanis is actually a really fantastic species for studying peak flowering time and how it changes over time. Because basically the way its biology works is that Triodanis will produce these closed flowers, the selfing flowers, early in its flowering season. Then in its peak flowering season, it produces those open outcrossing flowers. And then finally, in its late stage, like right before it dies, it produces a few more of the closed flowers. So if we look at herbarium records and find these open flowers, we have a good sense that we're actually capturing the peak flowering time, which is just, pretty useful if we're interested in seeing how the peak flowering time in a population has changed over time. All right, I'll get to my data. We found these are box plots of the peak flowering date from 1860 to the present. And we found this general downward trend, meaning that the day of flowering is earlier in Triodanis in the 2010s than it was a century ago. And overall, the flowering time has shifted about 12 days since the late 1800s. So we see this very strong shift. And I know you guys might be thinking like, okay, you have this map that spans, the continent, or spans the country at least, like from one side of the continent to the other. There is a ton of climatic variation in there. So how do we know that this is real when you consider all the other climate variation? Well, we built a multiple linear model. Our first one accounted for latitude, longitude, elevation, and their interactions. And even when we controlled for those geographic differences, we still found a significant relationship between year and day of year. And because obviously latitude, longitude, and elevation, they're not the full story. That doesn't encompass all the climatic variation that's available. We then leveraged PRISM data from the PRISM database, which is basically weather station data of many different climate variables, such as temperature and precipitation since the 1890s. And we were able to use principal components analysis to look at the relationship between climate variables and flowering day of year. And even when we controlled for all that climate variability, we still found that significant relationship between peak flowering and year through time, where peak flowering date was earlier in the later years. And so I'm just kind of describing that was our due diligence. It all checked out. That 12-day shift is real. But one thing that was really cool about looking into that PRISM climatic data and building that principal components analysis is that then we could pull out which specific climate variables were influencing flowering date the most. And I'm going to show you that data now. This is a pie chart from our principal components analysis, where these were the most significant variables influencing flowering time. And we used a metric. All of these variables, they represent a composite of March, April, and May for each of the variables. So if I say Tmax, it means Tmax in March, April, and May of that year. Most of the variance we saw in flowering date was driven by temperature changes. So basically mean temperature, max temperature, and minimum temperature in spring. And then a pretty decent sized chunk of the rest was actually driven by VPD, or vapor pressure deficit. You can kind of think of this like humidity. It has to do with the amount of moisture in the air and the potential for water to be pulled out of the leaves. So temperature and humidity were really important for driving this, and then precipitation played a role as well. So we found that this flowering time shift is influenced by these two factors, temperature and then the vapor pressure deficit. Now, we know that these things are changing through time, and we know that flowering time in Triodanis is changing as well. But our next question was about, like, okay, is Triodanis perfoliata even experiencing the stressful conditions, or is it able to just completely temporally avoid it by shifting its flowering time earlier? To address that question, we used a type of analysis called an ANCOVA, where this black line here shows the trend in March, April, May temperature over time from the 1890s to the present. It's going up because it's getting warmer. But this gray line shows the temperature during the time that Triodanis perfoliata is flowering. And basically, while one of these trends is going upwards, the gray is pretty flat, and it's even actually going downward a little bit. And so basically, Triodanis, while it is flowering, is experiencing pretty consistent temperatures from year to year because it is moving its flowering date. We found similar results for vapor pressure deficit. This black line is showing the trend in vapor pressure deficit over time. It's going up. That's a little confusing. Vapor pressure deficit going up means humidity is going down, so things are getting drier. But for Triodanis, while it is flowering, that stays pretty consistent. So what we find is that this is a plant that is pretty effectively able to temporally escape the stressors of climate change. Just to summarize, we had those three questions. Has flowering time changed? Yes, it's changed by about 12 days. Which climatic variables shape flowering time? Primarily temperature and humidity. And is it able to escape stressful climatic conditions? Yes, it is. It's successfully tracking the changing climate. And this concept of climate tracking, this is actually used to discuss sort of the ability of plants to sort of keep up with the changing climate. And there's these kind of interesting studies that look into whether a particular taxonomic group might be a good tracker or a poor tracker. And people are starting to sort of do research into what that means in terms of demographic consequences and ultimately species persistence. This data, it really effectively illustrates these changes through time across a range, but we still don't know whether these changes are plastic, genetic, or both. And I'm bringing back this image of all of the internal factors that help shape how a plant is going to respond to particular climatic cues. It's a pretty complicated question to try to figure out whether this change in flowering time is plastic or genetic, and what specifically is influencing these changes. But one way to investigate it is to look at related species and ask that question: is this broader taxonomic group kind of a good tracker or a bad tracker? One example of thinking about this in a taxonomic way, this comes from a study by the Willis Lab in Thoreau's woods. So Henry David Thoreau, he took really good records of the plants that were in the area around Walden Pond, and researchers have been able to return and conduct more surveys in the area. And they conducted this study where they measured how well these taxonomic groups were able to track climate change by altering their flowering time. And they also took demographic surveys to see whether a particular taxon was decreasing in population, increasing in population, or staying the same. And what they found was that many of the poor trackers actually had population decreases, and many of the good trackers had population increases in the time since Thoreau was at Walden Pond. I think this is really interesting to have this kind of phylogenetic perspective in looking at these studies. One thing, though, that I think is interesting as well is that Campanulaceae, which is the family that Triodanis is in, was actually listed as a group which was a poor tracker in Thoreau's woods. And at least in Triodanis, they're actually pretty darn effective trackers. And while I think this study is really cool, and I mean, overall, like very worth doing and doing studies like this, I also think it should be worth thinking about the fact that what is going on in one particular habitat does not give you the picture of what is going on in a species or in a genus or a family across its range. And so that's why I think it's really neat to kind of do these deep dives on a particular species across its range. So in sort of thinking about what we could do to examine the Triodanis genus as a whole, basically we just had a similar question. We said, okay, Triodanis perfoliata seems to be a good tracker. It is also a species that is quite flexible, has this huge range, is really weedy. What about the other species in the genus? And to do this, I teamed up with awesome undergrad at Southeast Missouri, Naomi Burr. She's since graduated. And we looked at the whole genus of Triodanis and asked similar questions of how flowering time was changing over time. Here's what we found. In 4 of the 7 species, we did find that flowering time was advancing through time. In 3 of them, we didn't. So it's not super conclusive whether all of Triodanis is a good tracker. It could be that for these 3 species, we just didn't have enough data to see the trend. I'd also like to comment that Triodanis leptocarpa, the trend looks like it's flowering later in the year, but this is just this relic of how much environmental variation there is. When I actually built the multiple linear models that accounted for the geographic variation, the trend was actually negative over time. So T. leptocarpa actually is flowering earlier when you account for all of the variance across its range. Yeah, so this was one approach that we used to look at whether Triodanis was sort of a good tracker across, and we found that 4 of the 7 species seem to be pretty good trackers. There's still, many directions that this could go in. The Weber Lab is doing a ton of really interesting research. Here, I recently saw the defense of Kate Tillotson's master's thesis, where she did this awesome survey of the pollinators of Triodanis, which is really relevant if we're trying to think of what influences Triodanis's decision to outcross versus self, for example. But yeah, so here's some other sort of future directions I've been thinking about for this is we know that Triodanis is advancing and it's tracking flowering time really well, but could there be a limit to flowering time advance? Like, is there any point where it might just get too early in the spring and the days are not long enough and it can't flower? That's still an open question. Then the question is, if Triodanis is so effectively tracking the climate, are its pollinators able to track the climate as well? Is there any potential for plant-pollinator mismatch when we think about this sort of advancing phenology? And then this is another kind of open question, is flexibility part of why Triodanis perfoliata specifically has been able to survive across such a wide range? It just seems flexible in so many ways, and I don't know. I think that's kind of neat to think like, oh, is this why you were able to colonize most of the Americas? Well, with that, I want to thank a lot of people. This research was conducted when I was at Southeast Missouri. And so, yeah, I want to thank, of course, Dr. Weber, and then many other sort of influences and mentors, and the sort of past and present members of the Weber Lab, as well as the herbarium networks throughout the US that enabled this research. And with that, I will take questions.